So good morning everybody, myself Waseem Akhtar, and today I am going to talk about the electrically detected magnetic resonance of phosphorus in silicon at low magnetic field. And this work has been done in collaboration with one of our PhD students, H. Morishita, and Professor R.S. Velasenko, Professor D.S. Ploskin, and Professor K.M. Ito. To begin with, I will just give an introduction of why EDMR is important at this era. So, as we all know that there have been lots of quantum computer models, and I list here the two basic models. One is Kaynes quantum computer model, and the other is all silicon quantum computer model given by our group. In both these models, we use phosphorus spin, nuclear or electron spin, as for calculation or readout purposes. So we want to detect the nuclear spin or the electron spin of phosphorus, and we want that it will be desirable to detect one atom or one spin, and for that we need a mechanism. We need a system which will detect one spin of phosphorus. So therefore, we look for some highly sensitive method, and EDMR was one of that method, which is highly sensitive. And to give you what EDMR is nothing but it is just a kind of a measure of electron spin resonance, where the change in electrical conductivity is monitored at the onset of resonance, spin resonance. I will explain it in more detail earlier. So, but just want to list the advantages of this method. That is, the signal intensity does not depend upon the frequency. That means that we can work at very low magnetic field without losing the intensity. And the second thing, it is highly sensitive than the conventional ESR. And there is a group here which reports that they were able to detect less than 50 phosphorus atoms in the sample. So this gives us this method, the electrically detected magnetic resonance, is more sensitive than ESR. Hence, a single spin detection needed for the realization of quantum computer is possible. As I told you that in electrically detected magnetic resonance, what we do is we measure the change in the conductivity of a sample at the onset of magnetic resonance. The change, the conductivity is generally given by this. So the change in the conductivity can be brought either by changing the number of carriers in your sample or by changing the mobility of those carriers. Now, if you work at a very high magnetic field and very low temperature, then the polarization of the donor electrons as well as the conduction electrons is very high, and in that case, the scattering term, the spin-dependent scattering, is really dominant at very high magnetic field and at low temperature. So, if you can play with the spin of the phosphorus, the donor atom, and the conduction electron, then you can control the scattering. And this scattering, the spin-dependent scattering, leads to change in the mobility. As a result, which will leads to change in the conductivity of the sample, which you can measure as a EDMR signal in your case. But there is another way to bring change in the conductivity, and that is by changing the number of carriers, that is N. And that can be done by recombination, and that is known as spin-dependent recombination. And today I will generally concentrate on this part because what we measure, our measurement, generally what we do is at very low magnetic field, where the polarization of the donor electron as well as the conduction electron does not play any significant role, as well as the temperature range which we work is from 5 Kelvin to 20 Kelvin, and this is high enough temperature where spin-dependent scattering is not at all dominant. So it is just the spin-dependent recombination which takes place, and I will explain the spin-dependent recombination in our case. Before explaining the recombination part, I just give you an overview of how phosphorus behaves in silicon. So this is under magnetic field. So this is the Hamiltonian, where this is the electron Zeeman term, you have the nuclear Zeeman term, and the hyperfine interaction term. In very strong magnetic field, you have 4 levels of phosphorus, where this shows the electron spin and this is the nuclear spin of phosphorus. So we have 4 levels of this. We can make transitions between ESR. Generally, this is the ESR allowed transition between these 2 levels. Then you have NMR allowed transitions, and then there are few transitions which is forbidden generally at high magnetic field, because these are not, because of the selection rules, these are forbidden transition. But now if you go to low magnetic field, that is below 200 Gauss, in this condition, you see that Now we don't no more have pure states, but a superposition state, something like this and this. So what was earlier a forbidden transition now becomes a allowed transition. I mean, you can see this signal. So we try to detect this signal in our sample, the forbidden transition, and we can also detect the ESR allowed transitions. So with this introduction to phosphorus, I will just show this is the experimental setup, and the sample what we use is natural silicon doped with phosphorus, and the concentration of the phosphorus I am using is 10 to the power 15 per centimeter cube. Then what we do is that we make electrical contacts for electrical measurement in our cell, and this is just the electrical the circuit for our measurement, and here is the cryostat which we use for EDMR measurement. Now, as I already told you that I will give you a brief introduction to how SDR proceeds in our phosphorus for detecting EDMR of phosphorus. Just give you a model of how phosphorus behaves here. So what we do is that first, for example, I will explain the energy levels here. This here is the surface center levels, and these are paramagnetic levels which is found at the surface of the silicon and the SiO2, interface of silicon and SiO2. So these are paramagnetic levels, which is also known as surface center or PV centers, and we have a donor level here. So what happens that when you illuminate your sample, then you create a hole and an electron. Now if this electron gets trapped here at this position, and they form a pair with this, the donor electron here and the surface electron forms a pair. Now if this pair is in singlet state, then it will recombine in a very short time scale, and then you will not see any change in the conductivity. But if it is in a triplet state, then it will not recombine very fast. So, but after application of RF, you can flip the spin of this electron, and then they can be in a singlet state, and then the recombination will take place, something like this. The recombination, the electron goes from here to here, and then eventually it will recombine with the hole. So what we see is that the conduction electron has been depleted of one electron here. As a result, the conductivity decreases, and that change in the conductivity you can measure as an EDMR signal. So I show you my results of phosphorus. You see that these are the two ESR transitions which I was talking earlier, and here are the signals of phosphorus. These two are phosphorus signal. In the center you have a peak which is from the surface center signal, and we are not much interested in detecting this, but today I won't be talking about these signals, but only about phosphorus. So we can detect phosphorus signals here, but the problem here was that we were not able to, because of the concentration of the phosphorus which we are using is just 10 to the power 15. So we were not able to see the forbidden transition, which is around 58. When you work at RF 200 megahertz, then it comes around near about 58 Gauss, the forbidden transitions. So the forbidden transition was not seen in these samples. Now I will just tell you what is the importance of these surface centers. To test the importance of surface center for detecting EDMR signal, what we did was that we treated our sample with hydrofluoric acid. So this black curve here is before the treatment, before treating the sample with hydrofluoric acid, and the red curve here is after HF treatment. You see that after HF treatment, the intensity of phosphorus, these are the phosphorus here, has gone down drastically. So this shows that when you treat your sample with HF, the paramagnetic centers decreases. The paramagnetic centers at the surface, that is surface center levels, decreases. As a result, the intensity of the phosphorus goes down. So to get a recombination, you should have higher concentration of PB centers or recombination centers generally. So for EDMR detection, recombination center is really important. But the problem in this sample was that here we were using the recombination center, PB center as our recombination center, but we cannot control the concentration of PB center at the surface. So we need something, some other recombination center which can be controlled. So we go for So we go for a gamma irradiated silicon. Because if you irradiate your sample, silicon sample with gamma rays, you can create lots of defects in your sample, and that defects will be deep level defects, and they can act as a recombination center for detecting EDMR measurements. So why gamma irradiated silicon? Because the defects created by irradiations has been widely studied, and there are a few defects in the samples which are known to be paramagnetic under illumination, and we need paramagnetic centers for recombination. And also we can control the defect density by controlling the dose of irradiation. With this, that gamma, I will just give you the brief introduction of what defect is important for our study is here. Generally, when you eliminate your when you irradiate your sample with gamma rays, you break the bonds here, and this two silicon will bridge with an oxygen, and here is a vacancy. So this oxygen and vacancy forms a pair, and this oxygen vacancy complex is one of the dominant defect produced under gamma irradiation. Now, if this defect is in a charged state, then it is known as A center, and if it is in a triplet state, something like this, and which is not charged, which is neutral triplet state, then it is known as SL1 center. Basically, I will talk about SL1 center here, and I will show here how the recombination takes place in SL1 center. What happens that when you illuminate your sample, just think about that we have these defects in our samples, oxygen vacancy defects, and here are the these two silicon have 1 electrons. So here I denote these two silicon, these two electrons. So when you illuminate your sample with a light, you create hole and electron. This electron will be trapped here, and it will go from a ground singlet state to an excited singlet state, or from a ground singlet state to an excited triplet state. Now, if it is in a ground singlet state, the recombination to the, if it is in the excited singlet state, then the relaxation to the ground singlet state will be so fast that you will not see any change in the conductivity. But if it is in the triplet state, then because we know that from triplet to ground singlet state, it is not allowed transition, so it will be metastable state here. But because of a spin orbit coupling, though very low in silicon, still we can see some transitions from the triplet levels to the singlet levels. Here I show you that in presence of magnetic field, this triplet level will be splitted, and they have different recombination probability from these levels. And if you play with around this using radio frequency, if you play around this, then you can change the recombination probability, and that will result in the change in the conductivity of the sample. So generally, this is what the signal will look like if you measure just the EDMR of SL1 center. It's something like this, that this here is the triplet state, this is the ground singlet state. Now if you play around with this part, if you play around along this using RF field, then you can see a change in the conductivity. We also see a change in the conductivity at these positions. These are basically crossing points, and you can see, and also at the zero field line, whose origin is yet to be studied. And here is one of the paper which talk about, in 1995 there was a paper which talk about detecting SL1 center, the electrical detection of SL1 center here, as well as they detected phosphorus. But the thing in the center, they talked about A centers and not PB center, which we were detecting earlier. So with this brief introduction, I will just come to these are the sample information where the irradiation time and dose of gamma irradiation. So I will this is the EDMR signal of gamma irradiated silicon. You see that, and this measurement has been done at a radio frequency of 400 megahertz. Now what we see is with phosphorus, these are phosphorus lines. These two are phosphorus lines, and in center we have a PB center line, which is surface center line. Now except this, we can also see 4 different signals, and there is one other signal here. This signal is independent of our radio frequency. And the thing is that This signal disappears when you go at very low modulation frequency, that is when we work at 1 kilohertz or so. This signal disappears. So we still don't know the origin why, I mean, at high modulation frequency this is observed. But I try to characterize this 4 signal studying the frequency dependence of the signals. So these are the 4 signals here, and this is the theoretical calculation based on this the Hamiltonian done by Professor Velasenko. So this is here I show that these 4 signals, 1, 2, 3, and 4, goes something like that. This is, I mean, the black one is part number 1, and this goes something like, I mean, it increases with frequency. So as we increase the frequency, the magnetic field also increases. So we are measuring something like here. So if you go on increasing the frequency, you measure somewhere here, and so it goes something like this. Now, if this signal is something that we see that with increasing frequency, the field at which the change takes place, conductivity change takes place, it decreasing. So we are measuring something somewhere here. So as you increase it goes on decreasing, and this part is constant, and this signal, which is this signal, and we measure this signal and as this part, the crossing point, which is independent of frequencies. And then there is another part here, the defect, which is at 90-degree orientation with the magnetic field, and this defect also contribute, and this is what we measure. This is what we measure here, which increases with frequency. So this characterizes, and so we can see that the experimental points as well as the theoretical calculated point matches exactly. So we can confirm that these are signals from SL1 centers. Now I will show you that in these irradiation samples, the sample which I studied earlier before irradiation, in that sample we were not able to observe the forbidden transitions. But here what we see that if we go and work at low modulation frequencies, we can see that we have phosphorus here, the PV center lines here, and there is this line here at 58 Gauss. We see that this line is the forbidden transition line. And we can see that even for 10 to the power 15 concentration, we can see the forbidden transition in irradiated sample. So this makes us to comment that because of the number of concentration of recombination centers in our sample has increased because of irradiation. Therefore, the recombination is much more efficient than the previous samples, and so we can even see the forbidden transitions in our samples. Now, if I compare my signals with the irradiated sample signals with the without irradiated sample signals here, you can see that there is significant enhancement in phosphorus level signal. So this enhancement is due because of the contribution from excess recombination centers which are created by gamma irradiations. So and I will explain the theory in a few other slides. Now if you come now, whether this enhancement in the signal is just because of because when you irradiate your sample, what happens is that you also create defects at the surface. So whether the enhancement in the signal is just because of the surface effect, or is it something, or we are having something from the bulk of the sample. So for conformity of that, we did the HF treatment results again, and what we see that after gamma irradiation, we measure the signals. So the signal intensity is something like that, and after that we do the HF treatment and then again measure the signal. What we see is that though the surface line has decreased, this is the surface line decreased, but the phosphorus remains more or less the same. So this makes us to comment that what we are getting is not from the surface, because and it is coming something from the bulk. So the radiation defects which are created in the bulk, they are responsible for the enhancement in the signals and not the surface defects. But this enhancement does not goes continuously with irradiation time or dose. Initially, we see an enhancement in the signal with increasing irradiation dose and the irradiation time. This enhancement is might be because of the overlap of the donor and the acceptor wave function. Acceptors our radiation defects act as an acceptor. So because of the overlap of donor and acceptor wave functions, you have a better recombination probability and efficient recombination probability, and hence the signal intensity keeps on increasing till some point, and then this enhancement is limited by the recombination time of the excited carriers. So even before you can flip the spins, they will recombine. So you will not see any change in the conductivity. So as a result, the intensity will decrease here. So this is limited by the photoexcited, the recombination time of the photoexcited carriers. Here I show the power dependence of the result of our sample. This curve is for the without irradiated samples. The red one is for the 20-minute irradiation and the 40-minute irradiated samples. What we see is that as we keep on increasing the irradiation time, the saturation is much faster. So exact reason might be, I mean, I'm not very sure about this till now. Might be that the concentration of carriers has to play some role in this saturation behavior. So if the concentration of the carriers are large, the recombination centers, if the concentration of the recombination centers are large, might be that leads to a better saturation. But still, the physics has to be studied still in a proper way. The other reason could be that this sample have, what we see is the recombination between the surface centers and the donor electrons. And the surface centers, these are paramagnetic centers, and they might lead to decoherence of the phosphorus electrons. But I'm not very sure, does T2 time of phosphorus has any effect on the power dependence? But still, there is a paper which reports that the T2 time is affected if you remove the oxide layer, that is the paramagnetic centers, then the T2 time does get affected. So might be if T2 time has any contribution to this, we think that the T2 time, if it has any contribution, that could also lead to a better saturation, a faster saturation. So still, it has to be studied in a proper way. Here I try to give a model of how does recombination is taking place and how SL1 center or radiation defects play a role in our EDMR measurement. What we do is that if you eliminate your sample here, you have the hole gets stabbed here in the SL1 center and just have one electron now, and the electron gets stabbed here in the donor site here. Now these two forms a pair. Now there is a possibility that this transfer of this electron to this level is a spin dependent transition, and that depends upon the spin of this hole here. So this transition from here to here, this level, could be a spin dependent transitions. And after that, this is the SL1 center, which we generally see in our results. We can also see the signal. So this is formed. Might be this can be formed. This state can be formed either by directly capturing the electron from here, or by transfer of donor electron or by the transfer of the donor electrons. So, and as we can see the donor electron also in our signal, so might be this transfer from donor to this level is one of the reason. Might be this is spin dependent transition. Other possible is the direct recombination of electron on hole at the donor site. I mean, you capture one electron, the second electron, and the hole is captured here, which forms a pair, and then they recombine. But this recombination process of direct recombination of electron on hole at the donor site is limited by the capture cross section of holes by the donor sites, and this capture cross section is not very high. So I don't know exactly how it contributes, what is the contribution of this direct recombination to the EDMR signal. With this, I would like to conclude. With this, I would like to conclude the talk. That is, in non-irradiated samples, we can the EDMR spectrum was attributed to due to the spin-dependent recombination between the donor and the paramagnetic centers at the interface. Now, we see a significant enhancement in phosphorus signals due to the spin-dependent recombination in the bulk between donors and the radiation-induced defects. The phosphorus EDMR spectrum in irradiated sample is observed in the range of this temperature range, which excludes the mechanism of spin-dependent capture of electrons by neutral paramagnetic phosphorus atoms, which is nothing but the scattering which I was talking about. Now, the EDSR forbidden transition between the phosphorus levels was observed in irradiated samples. With increasing concentration of radiation defect, the EDMR signal of phosphorus also increases, which is limited by the lifetime of the carriers.